moving into Chapter 8, we're ready to discuss microbial metabolism. This is a very high content chapter, so we're really going to focus on what is metabolism, what does that mean for microbes specifically, and really what the overall outcome of the different metabolic processes are as we go through the chapter. So first, let's talk about the term metabolism. When you think of metabolism, you should consider this to be all the different reactions that occur within the body. Metabolism encompasses reactions that break down molecules to give us energy. And so these catabolic processes are going to be things like breakdown of sugar, breakdown of fats, things that give us the energy we need to survive. The other side of the metabolism cycle would be anabolic processes. Anabolic processes will be reactions that take smaller particles and put those together. This will include the way we make fats to store energy, the way we make sugars when needed. These anabolic processes use energy. The majority of what we'll cover in this chapter will be the catabolic processes. We want to think about how microbes can take nutrition and break those different nutrients down to give them the energy they need to survive. Now, there are many different ways an organism can take in nutrients and break those down to produce energy. What we're going to spend the most time discussing will be the metabolic processes completed by our chemoheterotrophs. Chemoheterotrophs produces chemical energy by breaking down organic or carbon-containing compounds. So the chemoheterotrophs will be our organisms that take in sugars, fats, and break those down to produce ATP. And as you can see, this is the majority of what we will discuss. A phototroph, we have photohetero and photoautotrophs. These will be the organisms that can use light as an energy source. So these will be photosynthetic, and we'll discuss photosynthesis some towards the end of this chapter. Before we can begin discussing the pathways of metabolism, we need to become familiar with two different reaction components. The first one is our NADH and NAD molecules. These molecules have the job of carrying electrons around in the cell. Now, without going into detail on the chemistry of these molecules, let's take a look down here at the actual reaction. NAD is the oxidized form. That means it has the ability to accept an electron. So when you take NAD and you add electrons, you get NADH. NADH here is the reduced form. You can all kind of think of NAD as a little electron taxicab. Its job is to travel around your body, picking up electrons and carry them to where they're needed. The other important molecule for us to have a little understanding of is ATP. ATP stands for adenosine triphosphate. Adenosine triphosphate is our energy. What makes it energy for us is ATP has 3 phosphates. This last phosphate is connected by a high energy bond. So when you break this bond right here, releasing ADP, diphosphate, there is energy released in the process. So how would we make ATP? We would take the ADP, the adenosine with 2 phosphates, and stick this 3rd phosphate on. We only release energy when we break the bond. To make this bond takes energy. So our first pathway to discuss is glycolysis. Glycolysis is the catabolic breakdown of the most common sugar called glucose. Glucose, as you can see here in this drawing, is a 6-carbon compound. The process of glycolysis is 10 steps long. The first 5 steps, we actually have to put some energy in to get these molecules to split apart. Then in the payoff phase, we take our 2 molecules of 3 carbons, we pull some electrons out producing NADH, and then we release some energy producing pyruvate. So overall, we have taken the 6 carbon molecule of glucose, split it in half, and made it smaller, producing a 3 carbon pyruvate. The overall energy yield for this part of the metabolic process is 2 ATP, 2 went in, 4 came out, so we net 2 ATP, and we also get 2 NADH molecules that we can make energy from later. Glycolysis does not require oxygen. It can be completed by almost all microbes. Following glycolysis, the 3 carbon pyruvate will then have another carbon removed, releasing acetyl-CoA. Acetyl-CoA can then enter into the Krebs cycle. The Krebs cycle is a cycle where what we put in is exactly what we make at the end. The acetyl-CoA will combine with a molecule called oxaloacetate. So if you see here, acetyl-CoA is 2 carbons. You combine it with oxaloacetate, which is 4 carbons, making a 6 carbon molecule. We then keep popping through these reactions, stealing some electrons as we go. You see we make NADH several times. We make some ATP. We make FADH2, another electron carrier. We keep going until we ultimately end up back at that same 4 carbon molecule we started with. The purpose of the Krebs cycle is not to make the 4 carbon molecule. We had it to begin with. The purpose of the Krebs cycle is to steal or collect all of these electrons in the form of NADH and FADH2. So you can see our net energy yield from the Krebs cycle is 1 ATP, 3 NADH, and 1 FADH2. So how can we make energy from all these electrons we have stored up? We do this through oxidative phosphorylation. Oxidative phosphorylation is the process where we move these electrons through different proteins in the mitochondrial membrane, producing ATP. This is also called the electron transport chain. Now let me take this moment to point out that it depends on whether we're talking about eukaryotic or prokaryotic microbes as to where all of these processes take place. In a prokaryote, there are no membrane-bound organelles. So everything we've been discussing occurs within the cytoplasm. But if we're talking about a eukaryotic microbe where a mitochondria is located, at this point we would move into the mitochondria. But if this is a prokaryote, everything's happening in the cytoplasm, and we're just using the plasma membrane for this step. So let's look at how the electron transport chain works. If I use one of those little electron taxicabs, the NADH that has the electron, that NADH will donate its electron to the first protein. That electron then jumps. Every time the electron moves through these electron transport chain proteins, a proton, abbreviated here H+, those H pluses keep moving up. So we end up with a large amount of H+ on one side of our membrane. As those protons, those H pluses, flow back through this big protein called the ATP synthase, that gives us the energy or the movement that we need to produce energy in the form of ATP. Now this is a very simplified look at what's going on, but I want you to have an understanding of the fact that the movement of the electrons that we've collected from all the other reactions is what produces this gradient. The excess amount of protons flowing back down their gradient allows us to produce energy. So what do we do with these electrons at the end after they've been hopping through? Well, if this is aerobic respiration, those electrons will combine with oxygen and give us water. If it's anaerobic respiration, something else is going to have to collect those electrons. But oxygen is the most common final acceptor of electrons. This is why you need oxygen to breathe. And a lot of our microbes also need oxygen to live. But do all microbes use oxygen or always have oxygen? Well, the answer is no. So there is a backup called fermentation. With fermentation, this is going to be the process where organisms make some energy when oxygen is not present. There is no electron transport chain and no need to complete the Krebs cycle because we don't need the NADH molecules for the electron transport chain. Fermentation is glucose to pyruvate, so glycolysis, plus an extra step. So this drawing right here is just an example of one type of fermentation. So as glucose is converted to pyruvate, we get some energy. But remember, we also produce NADH. If we keep doing glycolysis over and over again to produce this energy, eventually all of our NADs will look like NADH. We'll need another step to take that NADH and grab those electrons to regenerate NAD to go back up and keep this glycolysis step going. The purpose of fermentation is this NAD plus regeneration. There are several molecules that can be produced at the end of fermentation. The two most common are lactate or lactic acid and ethanol. This is the way that a lot of your wines and alcohols are produced for consumption. They'll take a microbe, seal it off so no oxygen is present, and force that organism to complete fermentation. The organism will actually make the ethanol for us. So the overall key for those reactions was to an attempt to make energy. So now let's look at what would happen with our photosynthetic microbes. With photosynthesis, there's two distinct parts we need to look at. There's the light-dependent reactions and the light-independent reactions. With the light-dependent reactions, sunlight is used to help us produce ATP and NADPH. With the light-independent reactions, we'll use the ATP and NADPH from our first set of reactions to produce sugar. So looking over here at this picture, you can see on the left-hand side, we have the light exciting the electrons, producing our ATP and NADPH. Those molecules will then go over here to the light-independent reactions, enter into a cycle where we're producing our sugar source. So let's break that down a bit more. I've stolen a picture, an image from a different textbook to really explain this to you. So in detail, looking at the light-dependent reactions, there's two ways this can occur. We can have cyclic photophosphorylation, where the light will hit the chlorophyll, excite the electrons. The electrons hop through the electron transport chain. Remember, as that hopping through the electron transport chain occurs, we produce energy. Now, with the non-cyclic, we don't have the electrons hopping back to the chlorophyll. So you should kind of see that eventually that chlorophyll he's going to run out of electrons because as the light excites the electrons, they hop through the electron transport chain, and ultimately the electron goes over here to the NADPH. So chlorophyll needs some electrons stuck back in there. The electrons will come from water. Water is broken down, giving electrons to the chlorophyll and spitting out oxygen just as a byproduct. Well, as humans, we're really happy that that oxygen is released, but that oxygen is not helpful at all to the photosynthetic organism. This is really why we have to water plants. We all can easily think of plants as our photosynthetic organisms, but there are some microbes that are photosynthetic. They need water just like our plants do to replenish those electrons. So once we finish those light-dependent reactions, and we've made our ATP, and we've made our NADPH, then the independent reactions can occur. So we'll take some carbons, throw a whole bunch of energy in there, throw some electrons in there from the NADPH. Ultimately, we make sugar, which is how we've learned that photosynthetic organisms like plants can make their own sugar. They don't need to eat. So just to finish up this PowerPoint, let's go back through each process and point out the purpose. So purpose of glycolysis. Take that 6-carbon sugar, split it in half, preparing for the next step, making a little bit of energy as we go. Krebs cycle. Moving all of those carbons through the cycle to pull the electrons out. Why did we pull the electrons out? The electrons will move through the electron transport chain, producing our gradient to make energy. With photosynthesis, light-dependent reactions, light excites the electrons to move through the electron transport chain, producing energy and NADPH. Light-independent reactions, energy and electrons go in to produce our sugar. As you study, focus on the purpose of each reaction, what goes in, what comes out, and of course, understand the important molecules NADH, ATP, and understand the reason that fermentation must occur.